Welcome back to regulation of chromatin structure. In this video, you will be learning what is nucleosome positioning and what is the significance of nucleosome positioning. We told in the last session that nucleosomes are dynamic in nature. That is, the position of nucleosome in a DNA is not fixed. It can change its position from one particular sequence to another sequence. That is, they are dynamic in nature. But some nucleosomes are restricted in their position. That mechanism is called nucleosome positioning. This nucleosome positioning can be either by some proteins or by a particular type of DNA sequence. Here, the position of nucleosome will be fixed in a particular DNA sequence. It will remain as such. It cannot be changed from one position to another. Okay, so that can be mediated by either by the DNA sequences itself or by certain proteins. So first we will look what is the role of proteins in nucleosome positioning. Proteins act in two ways. In the first way, think that when a protein is already bound on a stretch of DNA, a second protein cannot occupy the same position. It is like a competition between two different types of proteins for the same DNA sequence. We know that when histones are present in nucleosomes, a regulatory protein does not have access to that DNA sequence. Similarly, if a DNA sequence is prior occupied by a regulatory protein, histone octamer cannot occupy the same position. Clear? So that is the nucleosome positioning mediated by protein. So think that when one protein preoccupies a DNA sequence, as given in the diagram A, represented by red colored protein, another protein given in purple color occupies a second position on the same DNA sequence near to the first protein, that is approximately 150 base pairs apart, less than 150 base pairs. In such a case, you can see two proteins bound on a linear stretch of DNA within a sequence of 150 base pairs. Here, the sequence in between these two proteins are inaccessible to histone octamer. Why? You know that the wrapping DNA sequence around the histone octamer has a size of 147 base pairs. So within 150 base pairs, if two proteins are preoccupied, a histone octamer cannot get the enough stretch of sequence to form a wrapping around the histone octamer. So that stretch of sequence will remain exposed. Okay. Then think that a third protein occupies next to any one of these proteins within another 150 nucleotide sequence. Then the sequence between these two proteins also will remain unoccupied by histone octamer. That means a great stretch of DNA sequence is nucleosome free. Okay. So this is the nucleosome positioning mediated by proteins first method. Okay. Here the protein binding prevents the formation of nucleosome. Okay. And in the second method, just see the diagram B, right side bottom. A particular protein represented by green colored structure has a high affinity to nucleosome structure. In this case, what happens if a protein binds to a stretch of DNA? That proteins will accelerate the formation of nucleosome near to the binding site of that protein. Here you can see the positioned nucleosome. This protein favors the formation of nucleosome near to its binding site. Okay, so that restricts the formation or accelerates the formation of nucleosome wherever that protein binds. So these are the two ways in which proteins mediate nucleosome positioning. Okay. Then we told that there is a second method by which nucleosome positioning occurs, that is by means of particular DNA sequence. We can have a look into that mechanism. Generally, a nucleosome forming region causes bending of DNA. Okay, you know DNA wraps around the histone octamer. Okay, so DNA has to bend over the histone octamer. So always histone octamer prefers the region which is capable of bending. Okay. In a nucleotide sequence, AT-rich region has a natural tendency to bend towards minor groove. Hence, when histones are facing towards minor groove, AT-rich region is favored. Okay. GC-rich regions are favored only when histones are facing away from minor groove. Got the point? AT-rich region has a tendency to bend towards minor groove. When histones are facing the minor groove, AT-rich sequences are favored. GC-rich sequences are favored only when histones are facing away from minor groove. You can see it in the left bottom picture. The minor grooves, whatever minor grooves are seen towards the histones are AT-rich, and the GC-rich regions are seen away from histone octamers. So these are the two mechanisms by which nucleosome positioning is mediated. Now, what is the relevance or significance of nucleosome positioning? Nucleosome positioning has both positive and negative effects. What is the positive effect? When a nucleosome is restricted or prevented forming in a particular sequence of DNA, that linear sequence of DNA remains exposed. That means any regulatory proteins can come and bind to that region. Generally, this nucleosome positioning is favored in transcription regulatory sites. Okay, So these regulatory sites always remain exposed so that regulatory proteins can come and bind to that region. This is the positive aspect of nucleosome positioning. Then what about the negative aspect? Negative aspect is that you can see that, for example, we told the second mechanism of protein mediated nucleosome formation. The position of nucleosome is restricted by the binding of a protein. Okay, that is, a protein favors nucleosome assembly to its immediate proximity. Okay, in such case, the DNA sequence which wraps around the histone always remains linked with that histone of taumer. It is applicable to all other nucleosome positioning also. In a positioned nucleosome, the DNA sequence is not free. It is not dynamic. It is fixed with the histone of taumer. So whatever sequences are present in that region of DNA will remain inaccessible to the regulatory proteins. Okay, so regulatory proteins are prevented from binding from these sequences of nucleotides which are involved in forming the nucleosome. Okay, so that is the negative aspect of nucleosome positioning. Okay, so these are about nucleosome positioning.